Welcome. Let me start by explaining the motivation behind this project. You probably have heard of the antimicrobial resistant crisis that we are currently finding ourselves in. More and more multi-resistant pathogens are emerging, while at the same time the rate of discovery of new antibiotics has decreased in the last few decades. We urgently need new antibiotics, but no one is certain where we can find them. Most of the antibiotics in clinical use come from bacterial specialized metabolites, and we know that the genes responsible can be found close to each other, forming biosynthetic gene clusters. Using genome mining, we set out to uncover if there is undiscovered chemical diversity in bacteria, and if so, where exactly. We started by collecting all 170,000 isolated genomes available from NCBI's RefSeq database, as well as about 50,000 metagenome assembled genomes from multiple projects. We mined them using anti-smash, the golden standard for rule-based BGC identification, and found more than 1 million gene clusters. However, it is possible that non-identical BGCs encode for the same compounds, therefore we needed a way to quantify their diversity instead of dealing with the pure numbers. We used two independently developed methods for similarity clustering. One is the published Big Slice tool, which is reducing the BGCs to their features using hidden Markov models from the PFAM database and comparing feature profiles to group the BGCs into gene cluster families. The other is the Clustomatic tool, which does the same thing but based on core gene alignments. The results of the two tools may differ on absolute numbers, but the conclusions we draw are the same. The effect of the similarity threshold is important here, and to confirm that our calculations are meaningful, we made sure that the GCFs that we find are as much as possible comparable to chemical classes. We ended up with around 60,000 GCFs. The contribution of the metagenomic data to this result is significant, as more than half of the diversity found in this dataset is unique to it. At this point, we were able to use the number of GCFs per genome, or MAG, to draw rarefaction curves, which allow us to estimate the growth of GCFs compared to the increase in sequencing data. Notice the dotted lines in the graph. The potential gene cluster families, the limits of said curves, indicate that there is a lot more to discover in the bacterial kingdom. We answered the first big question, so now to the second one. Where should people look for the next compound discoveries? To answer that, we needed to decide which taxonomic rank is most suitable for making comparisons of biosynthetic diversity. After an extensive variance analysis of all major ranks, we concluded that the genus is the highest rank with uniform diversity, so it can be used for prioritization and comparisons. In order to avoid any misclassifications known to occasionally occur in NCBI, we used the Genome Taxonomy database instead, which bases taxonomic placement on genome phylogeny and the metric called Relative Evolutionary Divergence, RED. However, for the purpose of staying reasonably close to existing naming conventions, GTDB accepts a range of values of this metric for genus definition. In order to make our biosynthetic diversity comparisons as fair as possible, we chose the median of this range of RED values as a cutoff and defined our own groups, the red groups. These are equivalent to genera but characterized by equal evolutionary distance among their members. At this point, we could quantify the biosynthetic diversity in each red group, but in order to minimize the sequencing bias, as some groups have been sequenced much more systematically than others, we drew again rarefaction curves with the purpose of comparing unevenly populated red groups. Since all the curves reach a specific number of genomes on the x-axis, we can compare those points, the potential GCFs, among the groups. For better visualization, we drew the GTDB's bacterial species tree, pruned to the red groups, and around it we plotted the bar plots of actual and potential GCFs. We can immediately see some interesting phyla and some very impressive outliers. This data is now public and available for download. It can be used as a guideline for future discovery efforts.